Hello students, welcome to the Crazy Science. I am your biology tutor. I am present here again with the new chapter of class 12th. So today we are going to start the molecular basis of inheritance, and in this chapter we'll be talking about the DNA, RNA, and about the histone protein, DNA proteins, genetic code, then different experiment which is done by the various scientists যে সেটা প্রুভ করার জন্য DNA is a genetic material or not. So this chapter is full of suspenses, lot of information are there in this chapter. তো এই চ্যাপ্টারটা আমরা জানি যে আমাদের বোর্ডসে and also in the NEET how much it is important. So it is carries huge marks. যেমন কি আমরা এক্সপেরিমেন্টস কিছু পড়বো in the upcoming classes where Griffith experiment পড়বো, Hershey Chase experiment পড়বো, then Melson and Staller experiment পড়বো. In every alternative year কিছু না কিছু কোশ্চেন্স ওখান থেকে আসতেই থাকে. So today I'm going to start this chapter with a very basic simple things. তারপরে one by one এক্সপেরিমেন্টে আমরা যাবোটাতে, ঠিক আছে? Let's begin today's class. So as you can see that I have given a headlines of the molecular basis of inheritance, and this is the most important chapter of class 12th. সো এই চ্যাপ্টারটার মধ্যে আমি আগে বলেছি যে ডিএনএ আরএনএ উইল বি দ্য প্রাইম, তাই তো? ডিএনএ আরএনএ উইল বি দ্য প্রাইম, অ্যান্ড ডিএনএ আমরা ছোটবেলা থেকে পড়ে এসেছি ডিএনএ আরএনএস আর কল্ড জেনেটিক ম্যাটেরিয়াল। সো জেনেটিক ম্যাটেরিয়ালটা আমরা জানি যে এটা কিভাবে কোত্থেকে এসেছে, কিভাবে হচ্ছে, নিউক্লিয়াসের মধ্যে যে ক্রোমাটিনস থাকে, ক্রোমাটিনটা ক্রোমোজোমে কনভার্ট হয়, and the chromosome we can to get to see the DNAs, and DNA আগে এলো কি আরএনএ আগে এলো সেটার ব্যাপারে আমরা জানবো। Okay, so DNA means deoxyribonucleic acid and RNA means ribonucleic acid. Both are called the genetic material of the cell. Both are called the genetic material. এই দুটোকে genetic materials বলে। Are two types of nucleic acid and found in living organism. যতগুলো লিভিং অর্গানিজমস আছে প্রত্যেকটা লিভিং অর্গানিজমসে আমরা দেখতে পারবো কি DNA and this two type of nucleic acids. DNA acts as a genetic material in the most organisms. বেশিরভাগ অর্গানিজমসে মেইন যে প্রাইমটা থাকে that is a DNA, and whereas RNA is also act as a genetic material in some organisms, in some viruses, and act as a messenger. So ব্যাপারটা হলো যে ডিএনএ ম্যাক্সিমাম those are the higher organisms. In those higher organisms আমরা দেখবো ডিএনএটাকে as a actual genetic যেটা থাকে না, যে actual genetic material হিসেবে, which is very important, and this is which carries the actual information from one generation to another generation, whereas the RNA, as you can see, this RNA যেটা থাকে এই RNA-টাও কিছু lower organisms, কিছু organisms এর মধ্যে RNA can act as a genetic material. For example, I can say the virus. We know the virus in mudde either DNA will be there, either you will see the DNA or RNA. ঠিক আছে? তো DNA থাকবে or RNA থাকবে। যদি RNA থাকে তো RNA can be the double stranded or the single stranded. তো সেই ব্যাপারটা DNA, we know the most important DNA যেটা স্ট্যান্ডার্ড থাকে ডাবল স্ট্যান্ডার্ড হেলিক্যাল স্ট্রাকচার দেয়ার লেডার লাইক শেপ, তো ওটা সবসময় ফিক্স থাকে ওটার মধ্যে। আচ্ছা, the DNA. It is a long polymer of the deoxyribonucleotides. A pair of nucleotide is also known as the base pairs, and length of the DNA is usually defined as the number of nucleotides present in it. Escherichia coli or E. coli is have the 4.6 into 10 to the power 6 base pair, that is BP. Haploid contain human DNA. These are the important information. 3.3 to 109 base pair, that is human DNA থাকে. In some cases, catalytic molecule. So basically, these are the two. একটা হলো E. coli এর মধ্যে তুমি কিরকম বেস পেয়ার দেখতে পারবে DNA, that is important, that is 4.6 into 10 to the power 6 BP. And whereas in the human DNA, যেটা human DNA থাকে, in the human DNA how much? এটা হলো 3.3 into 10 to the power 9 BP. এটা হবে 9, it's not 109, it's just a power. In some cases as a catalytic molecule. So these are the E. coli and human DNA. এটা হলো দুটো টাইপের এখানে বেস পেয়ার আমরা দেখতে পারবো, যেটা আমাদের molecular basis of inheritance এর prime information, and these two can come as a MCQ purpose. ঠিক আছে, আর বেসিক্যালি আমরা জানি যে ডিএনএ আর দে বোথ আর কল্ড দ্য জেনেটিক মেটেরিয়াল, দে আর দ্য নিউক্লিক অ্যাসিড। তো কি কোন একটা পিউরিন, পিরিমিডিন, দিস আর দ্য প্রাইম, দিস আর দ্য মোস্ট ইম্পর্টেন্ট থিং হুইচ ইজ প্রেজেন্ট ইন দ্যাম। তো ওই জিনিসটা আমরা নেক্সট স্লাইডে গিয়ে দেখছি। ঠিক আছে, অলরাইট। So in this is a structure of the nucleotide and nucleoside পড়বো। আমি একটুকে বললাম যে নিউক্লিওটাইড আর নিউক্লিওসাইডটা কি, একটা নিউক্লিওটাইড বললাম, নিউক্লিক অ্যাসিড যেটা বললাম, তো এখানে নিউক্লিওটাইড আর নিউক্লিওসাইডটা জানতে হবে। অ্যান্ড নিউক্লিওটাইডের স্ট্রাকচার পড়ে আমরা দেখব যে পলিনিউক্লিওটাইড চেইনটা কিভাবে ফর্মড হলো, যে ডিএনএ এর মধ্যে চেইনটা যে থাকে, দুটো ডিএনএ কে কানেক্টেড থাকে, তারা কিভাবে থাকে। নেক্সট আমরা দেখবো ডিএনএ আর বেসিক ডিফারেন্সটা কি, and the structure of the DNA will be talking about. After that, the packaging of the DNA, how they actually packaged, and up to এতটুকু আজকে আমরা ক্লাসটা করব। তো these are the most important যে কথাগুলো আমি বলতে যাচ্ছি, যে these all are the basic features of this molecular basis of inheritance. All right, okay. So in this one, so in this one structure of the polynucleotide, poly means बहुत, অনেকগুলো, nucleotide chains আমরা বলবো, একটা বলবো nucleotide আর আমি তোমাদের শর্টে বলে দেব about the nucleoside. Okay, so you can see, first of all go through this one. A nucleotide has three compounds, so we have to know that what are the three compounds we are going to talk about. A nitrogenous base, number one. Second of all, the pentose sugar and pentose sugar, ribose in case of RNA, and deoxyribose for the DNA, and the phosphate group. Okay, and the phosphate group. So these are the most three important components where the nucleotide is getting formed. One is a nitrogenous base, which is also called the N2 base, which is called the N2 base. Second is called the pentose sugar. Second of all, it is called the pentose sugar. Third of all, it is called the phosphate group. Third of all, it is called the phosphate group. Okay, so these are the three parts. One is a N2 base, second is a pentose sugar, third is a phosphate group. Okay, so these are the three components of the nucleotide. So তিনটে মিলে একটা নিউক্লিওটাইড ফর্মড হবে। Okay, so nitrogenous base is required, pentose sugar is required, and the phosphate group is required, and then nucleotide will be formed. All right, ribose in the case of RNA and the deoxyribose in the case of DNA. তো আমি যখন pentose sugar বললাম, তো আমরা জানি DNA আর RNA there is a big differences. তো DNA তে থাকবে deoxyribose sugar and RNA তে থাকবে only ribose sugar. The phosphate group, there are the two types of nitrogenous base, one is a purine and another is a pyrimidine. তো আমরা জানি যে nitrogenous base যেটা থাকে, একটা থাকে purine, another is a pyrimidine. So purine is something where the adenine, guanine, cytosine, thymine, আমরা ছোটবেলা থেকে since the childhood we are very known to this, right? So the purine is made up of the adenine, which is we call the ATGC, means adenine, thymine, guanine and cytosine. Whereas in the pyrimidine, যেটা থেকে দেখি আমরা, sorry, here one information I want to say, that is এখানে থাকে purine and pyrimidines are different. Purine থাকবে, in the purine you will get to see adenine and guanine, which is in short I can write ANG, whereas pyrimidine you will get to see that TC or U, means cytosine, thymine or uracil. ঠিক আছে, cytosine, thymine or uracil. AG is a common in both DNA and RNA. Both DNA and RNA তে AG টা common থাকে, whereas the C, T or U যেটা থাকে, it is a C যেটা থাকবে you will get to see in the DNA, and the C and U will get to see in the RNA. Means adenine, guanine both are common in the DNA and RNA as a purine. আর pyrimidine যেটা nitrogenous base-টা থাকে সেটার মধ্যে C means cytosine and T means thymine. Cytosine and thymine both are present. You will get to see where? You will get to see in the DNA, where cytosine and uracil U you will get to see in the RNA itself. Okay. So আমরা সবাই জানি DNA how they look. DNA is a double helical structure, right? Where RNA is just a single strand and responsible for the protein synthesis. And DNA that carries gene যেটা কি থাকে one generation to another generation, they are carrying the information. They're carrying the information. So this is how the phosphate group, nitrogenous base, all are they located to each other. They connected to each other. এরকম ভাবে থাকে। ঠিক আছে, তো data DNA, that DNA is carrying the gene or genetic information from one gene to another. ঠিক আছে, একটা জেনারেশন থেকে আরেকটা জেনারেশনে তারা এভাবে ক্যারি করে নিয়ে যাচ্ছে। Alright, so cytosine, uracil, I already said everything. Cytosine is a common in both DNA. C means cytosine, which is a part of the pyrimidine. It's a nitrogenous base, and thymine is present in DNA. Thymine যেটা থাকে শুধুমাত্র DNA থাকে বললাম, আর T only present in the as a pyrimidine. T যেটা থাকবে it is only present in the DNA, but instead of T, uracil U will be present in the RNA, which is a part of the pyrimidine. Pyrimidine is what? Pyrimidine nothing but it's a nitrogenous base. Uracil is present in RNA at the place of the thymine. ঠিক আছে, uracilটা প্রেজেন্ট থাকে RNA এর মধ্যে, and at the place of thymine, thymine জায়গায় uracilটা প্রেজেন্ট থাকে. This was a little bit idea of the nucleotide, which is also এরকম নিউক্লিওটাইড মালা থাকে, পুরো মালা থাকে in the entire DNA এর মধ্যে। এই ডিএনএটা ফর্মটা কি করলো, এই অনেকগুলো নিউক্লিওটাইড মিলে যেটাকে আমরা বলি পলিনিউক্লিওটাইড, একটা পলিনিউক্লিওটাইড মালা থাকে, ইটস লাইক কাইন্ড অফ বিউটিফুল এ মালা। তো ওটা মিলে পুরোটা ডিএনএ কে তারা সাকসেসফুলি বানায়। অ্যাকচুয়ালি ডিএনএ অ্যাকচুয়ালি লুক লাইক দিস। Okay, as you can see in the chart, nucleotide basically three types থাকে, that the component. One is the N2 base. This is a N2 base, nitrogenous base. Second is a pentose sugar. Third is a phosphate group. Okay, third is a phosphate group. For the nitrogen divided into two parts, one is purines and the pyrimidines. Purine means they are made up of adenine and the guanine, whereas pyrimidine is cytosine, uracil or thymine. So they are actually এরকম ভাবে থাকে। নেক্সট যে পেন্টোজ সুগার, একটা থাকে রাইবোজ, একটা থাকে ডিঅক্সিরাইবোজ। রাইবোজ you will see in the RNA itself, whereas deoxyribose you will see in the DNA itself. Okay, deoxyribose. Then phosphate group এখানে আর কিছু থাকে না। তো নিউক্লিওটাইড they basically look like this, and purine যেটা adenine and guanine, they both are present in DNA as well as RNA. In both you'll get to see DNA and RNA in both. Adenine, guanine, which is purine part of the nitrogenous base, they will be present. Where pyrimidine, cytosine, uracil and thymine together they are present, but cytosine and thymine you will get to see them in the DNA only. Okay, whereas cytosine and uracil you will get to see in the RNA itself. So এখানটা গিয়ে একটু just differentiation হয়ে যায়, other factor they are quite similar to each other. এবার কথাটা হলো যে আমরা জানো নিউক্লিওটাইড where they present. Now I'm talking about a nucleoside. So nucleotide থেকে যখন phosphate group বাদ হয়ে যায়, when there is no phosphate group, when there is no phosphate group means phosphate group gets deducted from which part? When phosphate groups get deducted from the particular nucleotide, this is called the nucleoside. This is called a nucleoside. Nucleoside means phosphate group won't be present. Okay, and phosphate group সেখানটাই থাকবে না, and phosphate group যদি না থাকে সেটাকে বলে আমরা nucleoside, and along with the nitrogenous base, pentose sugar and phosphate group, three of them together we say the nucleotide. And this nucleotide কোথা দেখতে পাওয়া যাবে? We'll get to see in the beautiful DNA itself. ঠিক আছে, এই ডিএনএ পুরোটাই পলিনিউক্লিওটাইড দিয়ে, একটা নিউক্লিওটাইড না অনেকগুলো নিউক্লিওটাইডকে একসাথে বলি পলিনিউক্লিওটাইড দিয়ে প্রডিউসড হয়েছে। অলরাইট, মুভিং টু দ্য নেক্সট স্লাইড। পলিনিউক্লিওটাইড চেইন, এতক্ষণ তো বললাম পলিনিউক্লিওটাইডটা হলো কিভাবে। নাও I'll be talking about the chain, how the chain is made up of, and এবার চেইন কি করে হলো সেটা জানবো। polynucleotide chain. A nitrogenous base, which is we call the N2, okay, is linked with the pentose sugar with the N-glycosidic linkage. Most of the cases erokom ashe, they asked in the examination. Examiner asked that N2 is connected with the, that N2 base is connected with the pentose sugar, right? So pentose sugar, if it is a DNA, if we are studying the DNA, we are experimenting the DNA, that means the pentose sugar will be deoxyribose, right? It's a deoxyribose. All right. So they are connected by which bond? They are connected by the N-glycosidic bond. They are connected with the N-glycosidic linkage. Okay, they are connected with the N-glycosidic linkage, means N2 is connected with the pentose sugar. ঠিক আছে, তো এটাকে বলে N-glycosidic linkage, দুটোর মধ্যে যে কানেকশনটা থাকে, this is called the N-glycosidic linkage to form the nucleoside. ঠিক আছে, এবার nucleosideটা ফর্ম হলো where the N2 and pentose sugar both are there. Now we require what? We require phosphate group. Phosphate group যখন এটার সাথে অ্যাড হয়ে যাবে, then it will become a nucleotide. Now they are nucleoside, cause as I said that nucleotide হতে গেলে you should have the phosphate group, you should have the nitrogen N2 bases, and also means purine, pyrimidine, adenine, guanine, cytosine, thymine, uracil, all you require, and you require the pentose sugar. If it is a DNA, you require the deoxyribose. If it is not DNA, RNA, they require the ribose sugar. So this is how the entire thing looks when a phosphate group is getting added and is linked with the 5' OH of a nucleoside through the phosphodiester linkage, nucleotide is formed. So as you can see, observe that there are the two linkage we are talking about. The linkage number 1 is glycosidic bond. This is a linkage number 1 and this is a linkage number 2. This is a linkage number 1 and this is a linkage number 2. So I'm talking about the two linkage over here. One is when the N2 is getting formed with the pentose sugar and the bond they are making, this is called the N-glycosidic bond, right? And here I'm talking about the DNA. If I talk about the RNA, the same, the pentose sugar instead of the deoxy, the ribose sugar will be formed there. So they are making connection. I'm talking about the DNA double helical structure, and here they are making the N-glycosidic bond. The N-glycosidic bond will be making the DNA as well as RNA in both cases. Where one phosphate group means phosphate group will only be present in the nucleotides, only nucleoside I'm talking about. Now, when the phosphate group will be added to this particular part and linked with the 5' OH of a nucleoside 5' OH, they will be present through the which particular linkage will be formed, phosphoester nucleotide is formed. So let's find out. This is a 5' phosphate and this is a 3' hydroxyl. Okay, so this is a 5' phosphate, this is a P, and hydroxyl means OH. So as you can see, there is a bondage ATGC, and ATGC, what they are the N2. They are the N2 bases. Okay, ATGC there, that means they are DNA. In the DNA, we can get to see adenine, thymine, guanine, and cytosine. There is no U, means no RNA. Okay. And they are making a phosphate. As you can see, these are the phosphate group they are making, means this is a proper nucleotide chain. Okay, and in between that they have a proper phosphate, they have a N2, and they also have the pentose sugar. The pentose sugar they also have in between that. তো এই প্যান্টোজ সুগার গুলো যেগুলো থাকে, এই প্যান্টোজ সুগার তিনটা মিলে তারা কি বানাচ্ছে, the beautiful connection of a making a their beautiful DNA double helical strand. আর ডাবল হেলিক্যাল স্ট্র্যান্ড যেটা আমরা বলি না, দেখতে যেরকম ডাবল হেলিক্যাল হয়, এইভাবে যে তারা ফর্ম থাকে, দে অল আর মেড আপ অফ দিস থিংস। এভাবে তারা যে কানেক্টেড থাকে, A T G C A T G C, and here the phosphate group is there, phosphate group is there. তো পুরোটার মধ্যে this is how their entire DNA is look like. Two nucleotides are linked, দুটো nucleotide, suppose this is a first nucleotide, this is another nucleotide, right? তো ফসফেট ফসফেটের সাথে যে বন্ডটা হচ্ছে, the phosphate phosphate এর সাথে যে বন্ডটা তুমি দেখতে পাচ্ছ, and this is called the phosphodiester bond. This is what I'm talking about. This is a phosphodiester bond. They are making a connection with it, linked with the 3 to 5 prime phosphodiester linkage to form dinucleotide. That's it. দুটো এখানে আছে, this is a number 1, this is a number 2, right? So they are making beautiful connection. One nucleotide is connected with the another nucleotide by making a phosphodiester bond, and here they are making in the form of 3 prime to 5 prime direction. 3 prime to 5 prime direction. All right? Okay. More nucleotide if they join, if the more than 2 if they are joining, that means they are making the polynucleotide. This is not more than 2. 2 means when 2 nucleotides they are making connection, but when more and more, there are 4, right? There are 4. When more and more nucleotide they just make a connection, they are making connection with the help of phosphodiester bond. So which bond is responsible here to making the two and this kind of, এখানে দেখছো এই দুটো এই চারটে এখানে ফর্ম, যে taking this kind of nucleotide in connected, they are called the phosphodiester bond. Okay, all right. More nucleotide joins together to form the nucleotide and polynucleotide. They are not more the di, they are poly now. They are poly means more than, they are more than 2. Okay, in RNA the nucleotide residue has the additional OH group present at 2 prime position in the ribose, and uracil is found at the place of the thymine. আমি আগে বলেছি, if you know, যদি তুমি এখানে কি পড়ছো না, if you are studying the RNA, in the RNA the cases will be different. ওকে, তো RNA তে কেসটা ডিফারেন্ট কি হবে, RNA এর মধ্যে you will get to see the uracil here, ইউ দেখতে পারবে instead of the thymine. থাইমিনটা শুধু থাকে in the where, in the DNA. ওকে, and also nucleotide যার মধ্যে additional তুমি একটা OH গ্রুপ দেখতে পাবে in the 2 prime position in the ribose and uracil, ribose sugar এর। So ribose sugar 2 prime position এ তুমি কি দেখতে পাবে, you will get to see what, the additional OH group. আরেকটা additional OH group, this is another speciality of the RNA. Okay, so additional একটা group you will get to see in that position. All right, moving to the next part. The structure differences RNA and DNA আমরা এখন জানলাম, the both most important the genetic material, and there are the 2 differences. We'll get to know these 2 differences. আমরা ছোটবেলা থেকে অনেকই পড়ে এসেছি about the differences. Now আরো বেশি ভাবে জানবো। The sugar present DNA is deoxy D ribose, whereas the sugar present is a RNA is a D ribose. We also know the deoxy because it's a DNA, and here's a ribose sugar, only ribose sugar is present. Second number, DNA contains the cytosine and the thymine. Cytosine means we call it C, and thymine means T we call, and as pyrimidine bases, and guanine and adenine purine bases. Guanine and adenine both are same in the DNA and RNA as well, but the main differences you will get to see in the which part, in the pyrimidine part. In this pyrimidine part you will be seeing the differences. In the RNA contains the cytosine and the uracil. In the pyrimidine bones, pyrimidine bases, guanine that is G and adenine that is A will be in the purine bases. So purine and pyrimidine both will be there. তো এখানে সেই ডিফারেন্সেসটাই আমি প্রথম থেকে বলে আসছি যে এডিনিন অ্যান্ড গুয়ানিন both will be the common, whereas the pyrimidine, cytosine thymine will be present in the DNA, and cytosine and uracil, that is C and U, will be in the pyrimidine. DNA has a double-stranded alpha helix structure, so they are basically double-stranded alpha helix structure, whereas the RNA is a single-stranded alpha helix structure. It's just a thread-like structure. It's just a thread. Okay, it's just a thread-like structure. All right. DNA molecules are very large. Their molecular mass may vary from, and this is not এখানে দেওয়া নেই, but আমি বলে দিচ্ছি, and RNA molecules are comparatively much smaller, with the molecule mass ranging from 2000 to 40,000. So DNA molecular massটা আরো বেশি থাকে, more than 60,000 এর মতো থাকে এখানে, ওদের মাসের যে ভ্যালুটা থাকে। RNA তে সবসময় স্মলার থাকে, 60,000 and 80,000 এর আশপাশ থাকে, 60,000 80,000 से ज़्यादा থাকে, কারণ এটা মাসটা বড়, and they are quite large in structure, and ওদের molecules গুলো অনেকটা বড় থাকে, 60 to 80,000 এর এরকম আশপাশে থাকে, whereas in the RNA molecule ওটা থেকে অনেকটা ছোট হয়, কারণ they are, you know that they are the double helical ladder like structure, right? And they also have the purine, pyrimidine, nitrogenous base, all of these things are there, and it is carrying the gene from one generation to the another generation. And RNA case এটা different, কারণ এটা just a single like একটা thread like structure থাকে, and only protein synthesis they do, শুধু protein synthesis টা করে, তাও সেটা থেকে ইমার্জ হচ্ছে, ঠিক আছে? তাদের ওই নাম্বারটা থাকে কম্পারেটিভলি ইজ এ 20,000 to the 40,000 মধ্যে তাদের যে মাসটা থাকে। ওকে, অলরাইট। So we'll be talking about the structure of the DNA, and then DNA helix packaging টা আমরা দেখবো, the entire process of it. So structure of the DNA, so basic knowledge you all have that they are just ladder-like structure, ladder-like তাদের দেখতে লাগে, double helix model for the structure of DNA, and it was given by the James Watson and the Francis Crick. They have this kind of scientist, that is Watson and Crick. এটাকে Watson Crick model বলা হয়, that is structure of the DNA কে Watson Crick model বলা হয়, and based on the X-ray diffraction data produced by the Wilklin and Rosalind proposed that this model of the DNA. So based on the X-ray উপর বেসড করে উইলক্লিন and the রোসলিন্ড এই দুজন মিলে ওদের কি দিয়েছিল, মডেলটার আইডিয়া দিয়েছিল, and also this particular one, the double helix, এটা ডিসকভার করেছিল ওয়াটসন and ক্রিক মিলে। Okay, there are the two scientist theory are included in this part. Next, salient features of this model. So I have just given 5 types of salient features of the DNA structure. Number 1, DNA is made up of the polynucleotides chain. আমি এখন দেখলাম না polynucleotides chain গুলা কি, multiple nucleotides when they connected to each other via the phosphodiester bond, in which backbone is made up of the sugar, phosphate and base is projected inside of it. So sugar and phosphate both are there. Sugar means deoxyribose sugar will be there. Deoxyribose when we are talking about the DNA here, to deoxyribose so থাকবে সেখানে। Next is 2 chains are anti-parallel polarity, আমরা জানি দুটো আলাদা আলাদা anti-parallel তারা প্রেজেন্ট থাকে, and one is A prime to 3 prime with the 3 prime to A prime. Okay, 3 prime to A prime, and one is a 5 prime, একটা থাকে 5 prime to 3 prime, আরটা থাকে 3 prime to 5 prime, তো উল্টো ভাবে তারা কানেক্টেড থাকে, ডিরেকশনে থাকে। The bases in these strands are paired through the hydrogen bond. So hydrogen bonded, that means adenine and guanine. If I say the ATGC, right? AT is connected with the double hydrogen bond, and GC is connected with the 3 hydrogen bond. So these are the bond which are connected, which are just connect করে রাখে দুটো nitrogenous base pair কে. This is called the H2 bond. Okay, these are called the H2 bond. Adenine and thymine forms the double hydrogen bond, just written here, and the cytosine form the triple hydrogen bond, with whom cytosine form the triple hydrogen bond with the guanine. So this is how the adenine and thymine এরকম ভাবে থাকে, and in this place adenine এর জায়গায় যদি এটা RNA হয় তো it will be uracil. Okay, hydrogen bond will be changed. Purine make a connection with the pyrimidine, and at the same time pyrimidine also make a connection with the purine, and here somehow hydrogen bond কোথাও দুটো কোথাও তিনটে এরকম ভাবে থাকে. All right. Fourth number is the two chains are coiled in the right-handed fashion. Right-handed থাকে, right-handed fashion এরকম ভাবে. Right-handed fashion means তারা connected থাকে, and the pitch of the helix is 3.4 nm and roughly 13 bp each around. তো 10টা bp প্রত্যেকটা রাউন্ডে 10টা bp থাকে, প্রত্যেকটা টার্নের মধ্যে 10টা bp করে they will be there. So the right-handed fashion they will be, the DNA will be produced. তো এই যে প্রত্যেকটা টার্ন যে তারা নিচ্ছে, প্রত্যেকটা টার্নে এই যে প্রত্যেকটা টার্নে 10 bps will be present, প্রত্যেকটা টার্ন যারা নিচ্ছে. ওকে, and also the pitch, যেটা pitch থাকে this is a 3.4 nanometer, এটা মনে রাখতে হবে. Fifth number, the plane of the one base stacks over the other double helix to confer the stability. So you can see that one helix is connected with the another helix, but these two helix ki korche ekta super ekta jate just lay down hoye ache for what, for more stability. And Francis Crick proposed the central dogma. It is the most important asked central dogma molecule in molecular biology, which states that the genetic information flows from DNA to RNA, RNA to protein. This is how DNA থেকে RNA form হয়, the RNA থেকে ultimate protein form হয়, and this entire process is called the central dogma, and it is proposed by whom? It is proposed by the Francis Crick. Okay, so Francis Crick, they may ask what is central dogma. It's something that's very important, like asks for the 2 marks. It is proposed by the Francis Crick, and it is something that central dogma ta ki eta bola hoy cause DNA to RNA, RNA to protein, means how the genetic information is flowing and ultimately ki outcome ta ber hocche otar upor এই central dogma is ultimatum. Okay, so these were the 5 type of basic features or salient features of the DNA. Now we'll be talking about the packaging of the DNA, how DNA is produced and get packaged, and what are the amino acids are responsible here. In the prokaryotics and eukaryotics, দুটোই পড়বো. Prokaryotics is very tiny. একটা information তো ज़्यादा দেওয়া হয়নি. Well defined nucleus is absent. We know the extent nucleoid is present. Nucleus is not there, only nucleate is there. Negatively charged DNA is combined with some positively charged, which is called the nucleoids. Okay, so prokaryotes ज़्यादा থাকে না, prokaryotes just nucleus থাকে না, except of nucleus. Instead of nucleus কি থাকে ওখানে just nucleoid is present, and nucleoid is where the nuclear membrane is not present. Nuclear membrane is absent. Okay, nuclear membrane is absent here. Moving to the eukaryotes, the histone is a protein. Okay, the DNA's protein is called the histone, and positively charged, always a positively charged protein, organized to form 8 molecule unit called the histone octoma. 8টা molecule নিয়ে produced, and this is called the octoma. And negatively charged DNA wrapped around it. So DNA always negatively charged. আমরা যে DNA জানি না যে DNA টা আমার double helical structure containing the information, they always the negatively charged. They always the negatively charged. Always. Okay, so negative positive মিলে একটা connection form হয়. তো এটা তুমি দেখতে পাচ্ছ that this is the entire histone protein, and the histone is a positively charged. Whereas this is a DNA, this is a negatively charged. This structure যে এটা দেখছো is just a double helical structure. Okay, they are the double helical structure they are forming at the end. So double helical structure তারা form করছে, DNA তে এটা form করছে, and it is wrapped by the histone protein itself. ঠিক আছে, এটা wrapped by the histone protein itself, as you can see. ঠিক আছে, এভাবে তারা বেরিয়ে যায়, DNA is just get out from the particular part. Okay, all right. So DNA and positive and negative mean histone are rich in this base amino acid, that is lysine and the arginine. May asked যে হিস্টোনের মধ্যে কোন অ্যামিনো অ্যাসিডটা ज़्यादा থাকে? One is a arginine, One is a arginine and another is a lysine. Alright, okay. Both amino acid residues carry the positive charges in their side chains. So, দুটোই এখানে কি করে side chains এর মধ্যে কি থাকে? They just carry the positive charges. The single nucleosome contain about 200 base pair, most important, chromatins in the repeating unit of nucleosome, and in nucleus some region of the chromatins are loosely packed and stains are light and they are referred as a euchromatin. Okay, তো এরকম ভাবে কি হয়, তো this is how they just wrap. Octamer means 8টে টাইপ অফ প্রোটিনস মিলে পুরোটা ফর্মড হয়, and 8টা টাইপের এরকম প্রোটিনস গুলো এভাবে একসাথে এইভাবে তাদের লুক লাইক দিস। ওকে, দে লুক লাইক 8টে টাইপ অফ প্রোটিনস মিলে তারা এই অক্টোমারটা ফর্মড হয়, and this octamer is responsible for making the DNA strand. তো আলটিমেটলি ডিএনএ স্ট্র্যান্ড গুলো হয়ে পিছিয়ে থাকে না, তো ডিএনএ স্ট্র্যান্ড গুলো হয়ে তারা বেরিয়ে যায়, ঠিক আছে। and this is what nucleus এর মধ্যে some reasons chromatin as loose কিছু কিছু জায়গার মধ্যে chromatins না just loosely তারা থাকে, they are not tightly packed, they are just loosely packed, and they are referred as, and they also stained light, অনেক লাইট স্টেইন থাকে, and this is called the euchromatin. This is called the euchromatin. We know the chromatins are present where? In the nucleus. Whereas the chromatin that more densely packed and stains more dark, this is called the heterochromatin. And you can see that here is just a network of chromatin. This is called the heterochromatin. This is called the heterochromatin. And it is quite dark in color. Okay. The euchromatin is transcriptionally active chromatin, and whereas the heterochromatin is a inactive. So euchromatin যেটা থাকে means this is a euchromatin very active because this is a white light also. এটা একটা active chromatin থাকে, but যে heterochromatin খুব densely থাকে, they are quite dark stained, they are not that much active, they are inactive all the time. Most of the information is carried by the light. এটা কোথায় থাকবে? It will present in the nucleus এর মধ্যে থাকে, in the nucleolus we get to see, and it also present in the nucleolus and nucleus both where, especially nucleus এর মধ্যে তোমরা দেখতে পারবে this kind of structures গুলো. So when positive and negative both are connected, make a beautiful connection in the DNA, and in the DNA we'll get to see ki euchromatin, heterochromatin. So this is how the entire package of the DNA or DNA helix jeta hoy, means double stranded DNA helix, it gets formed. Okay, in the next class we'll be talking about the another experiment, the Archie and Griffith experiment. Till that, stay tuned with the Crazy Science. Keep studying, keep working hard. Thank you.